Good morning, everyone, and welcome to the 2025 Reefs Go Live series. We're broadcasting live today from a dive site known as Windsock Reef here in Little Cayman for our first episode of the year. My name is Balt von Huyn. I'll be your topside host today. Joining me as our underwater host is Chandra Delpeche. She's wearing a specialized face mask that lets her both hear and talk underwater. So let's say hi to her now. Hi, Chandra.
Hi, everybody. I'm so happy that you could join us today. Welcome to the first episode of Reefs Go Live. We're going to be talking to you today about underwater seamounts and why they're so important. So before we start, we want to tell you guys or encourage you all to leave your comments and questions in the chat. And we will be able to try and answer as often and as soon as possible. To kick things off, so tell us where you're from and if you've ever heard of seamounts before.
Thanks, Chandra. Looks like we have viewers from all over the world joining us today. Now, we also have a dedicated team from CCMI working behind the scenes, both on the boat and underwater, to make today's episode possible. So let's give them a shout out.
Hello.
Now, I have here in the chat in front of me some viewers. Bob from Hampton Roads, Virginia is joining us. James from Michigan, Triple C School in Grand Cayman. Jerry from Cayman. Gary from Abilene, Texas. Cindy from Bluffton, South Carolina. Village, Mount Catherine from back home in Arousic, Maine. Denise from Tallahassee. Pedro from Portugal. George and Mia from Program Beach. Austin, Texas. Karen in West Bay of Grand Cayman. Yvonne from France. Dottie from West Bay, Grand Cayman. Christine from the UK. Kim from Georgia. Great to have so many of you with us here today. Now today we want to keep hearing from you. So as you can see, I can see your comments in the chat in front of me. So send us any questions or comments you may have. We'll try to reply to as many as we can. Meanwhile, Chandra, would you like to introduce today's topic?
I would love to. So we're going to be talking about underwater seamounts. So underwater seamounts are basically underwater mountains, quite self-explanatory. And they are formed through volcanic activity. Now the special thing about seamounts is that they are host to mesophotic reefs. Now mesophotic reefs, compared to our shallower reefs, we find them in a depth range of between 30 and 150 meters. And they are host to all the organisms that can thrive in low light conditions. They're also known as biodiversity hotspots. So we would like to hear from you guys, the viewers, if you know what a biodiversity hotspot is. Charts in the chat, so later on the episode we'll come back and see how many of you got it right. In the meantime, Bolt, can you tell us why seamounts are so...
I'd love to. A seamount in the open ocean is like an oasis in the middle of the desert. If you think of the sunlit layer of the open ocean, it's hard for even a lot of marine life to live there. There's nowhere to hide from predators. There's no places for things like coral or sponges or seaweed to grow. But if you have a seamount, that adds structure and that adds habitat for all kinds of marine life. And that allows vibrant ecosystems such as coral. And at the same time, a lot of nutrients out in the ocean. This brings nutrient-rich water to the surface and allows tiny organisms called plankton to thrive. And those plankton in turn feed everything from small fish to sharks and whales. Now, makes seamounts crucial to the health of our ocean's ecosystems. Chandra, can you tell us a little more about how these seamounts form?
So, I've mentioned before that seamounts form through volcanic activity. This is when magma from the Earth's mantle pushes up through the ocean crust and it forms these underwater volcanoes. These underwater volcanoes later on grow into seamounts. Now, this is important because because they're biodiversity hotspots. So while Bolt is checking, if you guys know what the answer was, if you've got any questions right, or the question right, rather. A biodiversity hotspots are regions in the ocean where it has a high species diversity. And this includes all your endemic species. And within these endemic species, it will also include a number of endangered and threatened species. Bolt, our response is looking from up top.
Well, let me have a look here. I see Village Montessori is submitting a biodiversity hotspot, lots of sea life. And that does capture what a biodiversity hotspot is, as Chandra just mentioned. Now, so nicely done. Chandra, a little bit about the geology of seamounts, which itself is a really interesting topic. Scientists spend a lot of time studying the rocks and sediments of seamounts to learn about things like past volcanic activity and the movement of Earth's tectonic plates. But seamounts aren't just geological features. They are vibrant ecosystems that are packed with life. And the biodiversity found on seamounts is often much higher than that of the surrounding ocean. And this, again, is because the structures of seamounts provide these numerous niches for different species to thrive. I'm curious, what kinds of sea creatures do you think you might find at a seamount? Go ahead and send some guesses into the chat, and I'll check the comments as we go. Now, meanwhile, Chandra, can you tell us more about the biodiversity on seamounts?
Sure. So, there is a wide variety of diversity on seamounts. You can find anything from sponges and corals to the tiniest crab and the largest sea mammal. Now, a lot of these animals, or a lot of the biodiversity, is an endemic species, and that's why it shows the importance of conserving these underwater seamounts. When it comes to the biodiversity, and why, there are a couple of reasons. The first being that it's a nutrient-rich environment, and that there's abundance of plankton, and this plankton abundance forms the basis of the food web. All this plankton then attracts a lot of other fish, like larger mammals and all your predatory fish. Another reason why the biodiversity is so high is seamounts have a high structural complexity. So this basically means that there are a lot of nooks and crannies for fish species hiding.
That's right, Chandra. Seamounts are like underwater cities. They're bustling with life. They are also vulnerable to human activities. For example, they often provide good fishing grounds, and therefore become overfished. And things like deep-sea mining can actually destroy those critical habitats that Chandra was talking about. Meanwhile, human-induced climate change can actually alter ocean currents and temperatures, and it can destroy life. It is therefore crucial to protect these seamounts to maintain the health of our oceans. And marine protected areas, or MEAs for short, are one way to preserve biodiversity and ecological functions by restricting some damaging human activities. Now, Chandra, can you explain more about how MPAs work?
Sure, so MPAs, as you've mentioned, are these regions in the ocean where human activities, such as fishing and mining, are limited or not allowed at all. And the whole reason for this is to protect and preserve marine life.
Exactly, that's a great explanation. MPAs can be created by governments, international groups, even local communities, and they are essential tools for ocean conservation. As a matter of fact, we're sitting in an MPA right now, the Preston Bay Marine Park on Little Cayman. But MPAs can't be the only solution. There are other important steps that we can and must take to protect our oceans. And this includes things like supporting sustainable fishing practices, responsible tourism, and reducing our carbon footprint. And we'd love to hear some ways that you all think of to help protect our ocean. If you would like to leave those in the chat. Speaking of the chat, let me look at some of the comments we have coming in and see if there's any questions that we should answer. I'm seeing a lot of good responses to what a biodiversity hotspot is. That's encouraging. People joining us, Daniel from South Africa. I believe Andre Delpeche may also be joining us from South Africa. Great class from Michigan. Nancy from Vancouver. Great to have you all with us. Oh, and a happy birthday to Fiona Mosley. I'm seeing here a response from Village Montessori. Beach cleanups, limiting plastic consumption as ways to protect the health of our oceans. Dario says, don't overfish. All excellent and accurate responses, so thank you all. Now we've covered how MPAs work. Let's consider the larger context of why they're important and why it's important to protect these seamount ecosystems. Jay, can you tell us more about the impact these seamounts have on the health of the wider oceans?
Well, seamounts play a massive effect on the oceans. They affect the way the ocean mixes and cools. And this in turn affects how they heat and store carbon. This is really important because it can act as a buffer for climate change. Another important effect is that seamounts can be used as stepping stones for population or species migration. This is benefactory because if there is, changing ocean temperatures and a rising ocean temperatures, species that need to move to cooler ecosystems are able to hop to these stepping stones or seamounts, and this also helps with connectivity of population density.
That's right. And seamounts also offer refuge because many seamounts are out in the middle of the deep ocean. And that protects marine life from the impacts of things like coastal development and pollution. But they can still be threatened by activities like deep-sea fishing and mining. And this is why it's crucial to study and to protect them. And this is why CCMI's research team has been so invested in a recent project called Expedition Hope. By understanding and protecting these ecosystems, we can help promote a sustainable future for modern oceans. That's what Expedition Hope is all about.
Beyond the Cayman Islands, hidden beneath the surface of the ocean, lie towering underwater mountains, uncharted and unexplored, until now. Through Expedition Hope, our scientists here at CCMI spent 3 years researching 12 Mile Bank and Pickle Bank, braving unpredictable waters to document never-before-seen ecosystems and the forces shaping these deep-sea habitats. Their discoveries revealed healthy, diverse coral communities, home to a variety of marine life, including species rarely seen in shallower waters. The findings challenge what we thought we knew about how reefs survive stress, and may help us understand how some ecosystems adapt to climate change. With Expedition Hope, CCMI aimed to expand knowledge, support conservation, and raise awareness of these little-known ecosystems. By studying this environment, we hope to help protect our oceans for the future.
Now that was a great introduction to this project. Chandra, can you explain a little bit more about...
Namely Pickle Bank and 12 Mile Bank. This program was funded by the UK's government Darwin Plus program. Would you be able to elaborate on our field expeditions that we did?
Absolutely. The objective here was to document both the benthos and the biodiversity of few offshore seamounts, which were previously more or less unexplored. These being 12 Mile Bank and Pickle Bank. In August of 2023, CCMI's research team joined with researchers from the University of Delaware, as well as Harvard Oceanographic Institute and the Smithsonian Tropical Research Institute, to explore these poorly documented reefs on 12 Mile Bank. This is located 12 miles off the west coast of Grand Cayman. And they surveyed the biodiversity of the reefs there, documenting both fish and coral diversity at depths of up to 50 meters, a little over 150 feet for viewers in the U.S. And last summer, they repeated this project at Pickle Bank, which is an offshore seamount 45 miles off the coast of Little Cayman. And these surveys will aid in the better management of these important but, again, little-known ecosystems. Now, Chandra, can you tell us what's currently on seamounts?
Of course. course. So right now, we know that 12 Mile Bank and Pickle Bank are used for recreational and commercial fishing. We, however, don't know the link between the species on seamounts and in the general Caribbean.
And that's why this project attracted researchers from a variety of institutions along with CCMI. And some of the collaborators that brought both resources and expertise to this project included the Cayman Islands' own Department of the Environment, the Ocean Foundation, and again the Smithsonian Tropical Research Institute. And the result was that this team was able to use some fairly advanced technology to survey the marine life of these seamounts. For example, one important technique is known as eDNA analysis. Andre, can you explain a little more about eDNA analysis?
Well, it is first important to know what DNA is. So DNA is a genetic makeup that tells us who and what we are. Now, eDNA is environmental DNA. Now, what happens to environmental DNA? When marine species swim around the water, they shed their cells, mucus, and gills. And this is what we as scientists, and now you the viewers, know as eDNA. This is important to our researchers, and it will tell us exactly what organisms or species are in environmental surroundings, even the ones we cannot see.
That's right. And eDNA is vital to the success of this project. Other examples include the development of what are called benthic photomosaics. These enable our researchers to analyze the structure and the composition of a reef later from photographs they took during their surveys. And our team actually had great success with this technique. They completed 20% more photomosaics than they had planned. And this project was only able to accomplish all of this with multi-year funding recently awarded by the Darwin Initiative, as well as with the support of an anonymous private donor. And the results of this project will be useful for conservation. It will also add to our understanding of the importance of seamounts worldwide. Now, that being said, not every single aspect of this project was so high-tech. So, Chandra, can you tell us a little bit more about how they conducted their fish surveys?
So, you can see I've got some of the tools down here with me. What our researchers like us did is they took what we know as transect tape, which is this thing over here, and they were able to lay out a transect line. Now what they did, I'm just going to put that down for a second, is they swam on either side of this transect line, and they identified and counted individual species. Using this T-bar, you can see there are increments, and each increment is marked in 10 centimeters. This helped the scientists estimate the size of the fish that they were spotting. And they were able to do this all with use of rebreathers. Bolt, are you able to tell us what a rebreather is?
I'd love to. I'm going to take just a quick moment to address some questions I'm seeing in the chat here. We have a question regarding the volcanic activity of these seamounts. If the volcano would again, would it hurt the fish and plants? And to my knowledge, neither of the seamounts being studied here, 12 Mile Bank or Pickle Bank, are considered to be active volcanically. I could be wrong. I'm actually not an expert on that at all. There are underwater volcanoes that are erupting, some continuously, some more violently, and that's actually a really interesting topic, is what sea life is able to live around these underwater volcanoes. In some cases where the eruptions are not so violent, the more continuous release of volcanic gases and material, they actually create a local hotspot in and of itself, in that case literally a hotspot. Things like thermal vents, where communities of fish and other organisms live around them, essentially don't live anywhere else. So if you want to learn more about that, Google thermal vents. It's a really interesting topic. I'm going to talk for a moment about what Chandra mentioned, which is rebreathers. Scientific divers on this project were using rebreathers. A rebreather is a device that essentially replaces a conventional scuba tank that you're wearing back. It actually recycles the air that you are breathing underwater,
instead of just supplying compressed air
from a scuba tank. And this has two very important advantages. First of all, it allows divers to stay underwater longer, so they can get more work done. And it also avoids releasing bubbles into the water. One issue with surveying fish using a scuba tank is that the bubbles, as you exhale and breathe out, actually disturb the fish sometimes and scare them away as you're trying to count them. So by using these rebreathers, this research team was able to get much more accurate and thorough fish counts and observations. Now, Chandra, can you tell us a little bit more about some of the other methods they used?
We've spoken about the fish surveys, and another survey they did is the benthic survey. So what our researchers did with benthic surveys is they took cameras underwater and actually got to take pictures of the benthos, or the ground. Eventually, they stitched all these photographs together to form a photo mosaic. Now with this photo mosaic, they were able to identify all the different hard corals, and they visually assessed the sponges, algae, and cover. What else do you think, Bolt? Can you have any thoughts on this?
Yes. One other interesting thing I'll add to that is from these photo mosaics that Chandra is describing, our team used some very specialized software to develop a three-dimensional computer model of what the seafloor looks like, what the benthos looks like. And these 3D models are actually game changers in marine research.
because being able to view those complex structures on the seafloor in 3 dimensions is very important for understanding
how benthic organisms are distributed, and therefore how to better conserve and manage these ecosystems. Now let's have a quick look at this project's results. At both 12 Mile Bank and at Pickle Bank, our researchers discovered very productive ecosystems and very healthy coral reefs compared to Cayman's injuries along the coastline. Now this is likely due to their remote location as well as their slightly greater depth, and both of these offer some protection against human impacts and coral bleaching events.
Now, the fish surveys have revealed some fascinating insights into the fish communities of these
reefs. Sites, for example, showed significantly higher fish density and diversity. This was likely due to stronger water mixing at those sites, again, higher nutrient availability, as we mentioned earlier. Dominant species at these sites include things like basslets, damselfish, wrasse, and chromis. There are also larger fish such as snapper and parrotfish also accounted for a significant amount of the biomass, even though their numbers may have been low.
The researchers also observed 2 mesophotic specialist species, the black-capped basslet
and the sunshine fish, which were abundant on the seamount but really not present on shallow reefs. Now, Chandra, here's a challenge for you. Say the word sunshine fish 10 times fast.
Just let me give it a try. Sunshine fish, nope, I definitely cannot do that. Well, based on the results you were telling us, it's also important to note that invasive lungfish were barely seen on these seamounts. So it's kind of indicative that they are not yet a major problem on the seamount ecosystems. Can you tell us a little bit more about the benthic habitats?
I'd love to. I'm going to pause for just a moment to address a question I'm seeing in the chat. What is fish density and diversity? So let me clarify a little bit. Fish density refers to how many fish do you see in a given area, right? How dense packed are they? How many do you find in a
area on the reef?
Fish diversity refers to the number of species. You can have a lot of fish, but if they're all one species, that's not a diverse ecosystem. So a diverse reef will have
Multiple different fish species represented. With that clarified,
let me talk about habitat, as Chandra alludes to. In terms of the benthic habitat, and again, the habitat along the seafloor at these seamounts, at a depth of, say, 45 meters, coral cover averages about 2.6%. That's live coral cover, as a percent of the total area, which is similar to other mesophotic reefs in places like Honduras and Mexico. Slightly lower coral cover compared to places like Dominant coral species here include things like lettuce corals, star corals, and boulder star corals.
Chandra, can you explain how it's even possible for these corals to survive on mesophotic reefs?
Sure. So that's a really interesting topic. But I think we first need to understand how corals feed. There are two main ways that corals feed. The first one is they have this relationship with a symbiotic algae known as zooxanthellae. And the zooxanthellae photosynthesizes using light, right? And this provides the coral with about 90-95%, or majority of its nutrients. Another way the coral feeds is using its tentacles, and it uses tentacles with a sticky mucus net to catch tiny organisms that float in the marine ecosystem. So, some adaptations that these corals have to living these deep, dimly lit environments is they grow wide flat leaves. This increases their surface area, which enables them to photosynthesize. Another adaptation is that they grow slower. So this slow-growing coral enables them to be more tolerant to warmer temperatures. And this is probably beneficial with our warming oceans, right, Bolt?
Absolutely. Another interesting point that I'll bring up here is the role of sponges in these ecosystems. Sponges are actually the most abundant benthic invertebrates. They make up over 10% of that benthic.
And this supports previous observations that show that sponge diversity actually increases with depth.
More up to a point at least. And sponges play a crucial role in ecosystem health by promoting nutrient cycling and also providing habitat structure. So who would have thought, Chandra, that sponges of all things are so important?
I know, it's amazing to see how everything is so interconnected. And this highlights the importance of how we need to learn exactly how the marine environment is changing so that we can protect it adequately.
Exactly. Now, these previously unexplored seamounts could provide a glimmer of hope for Cayman's coral reefs, potentially providing refuge and resilience from the impacts of climate change. And the results of Expedition Hope are a huge step forward in better understanding and therefore protecting these valuable ecosystems. Now let's check in with our dive team briefly before we answer some questions. How's everybody doing down there, Chandra?
We're having a swell time, Bolt, so I'd love to hear what our viewers have to say.
Excellent. Let me take a quick look at what comments and questions we have coming in.
Let's see. First of all, hello to Charlotte from LaGrange, Georgia.
Doug asks, how much stony coral tissue loss disease are you seeing in Little Cayman? We have input from other team members here saying disease isn't confirmed in Little Cayman. We did, of course, see excessive coral bleaching in 2024. Also 2023, it was the marine heat wave, during which we lost about a half the coral cover around Little Cayman Island.
Fortunately, most of the key reef-building species recovered after that bleaching event.
So to Doug, I guess I would say we can't really say whether what we're seeing is stony coral tissue loss disease, especially relative to how it behaved in Grand Cayman. I don't know. Some of your viewers may not be familiar. Stony coral tissue loss disease was and still is, a devastating coral disease that's impacted a lot of the reefs around the Caribbean. Grand Cayman, for example, suffered greatly. A lot of the large corals there got infected and died. It's a very fast-moving and fast-acting disease, and it's something that a lot of scientists and
Conservationists have been very concerned about in terms of the reefs on Little Cayman. For now,
it doesn't seem to be having the same impact here. Again, I myself am not an expert on it, so that's as good of an answer as I can give that question. I have a question here from Lily May. Do you like working in marine science? Oh, I think all of us here love working in marine science. It's really a dream come true, I think, for a lot of us, and I would encourage anyone interested to pursue options for working in that field as well. Hugh asks, any plans for studying the deep sea mounts in the Bogue between Brack and Little? Not that I know, but that's a very interesting point. I know very little about the bathymetry of the Bogue. A fascinating stretch of water. For those of you who aren't familiar, Little Cayman and Cayman Brac are the two sister islands in the Cayman Islands, located, I think, about 5 miles apart where that name comes from. Hugh here is alluding to the presence of deep sea mountains in that stretch of water. Interesting question. I wish I had an answer for you. I'm going to look that up myself when I have the chance. The students at CPCH want to know what your jobs are. Chandra, would you like to talk about your role at CCMI?
Oh, sure. So, I'm an education assistant at CCMI, which means I get to work with all the education groups that come through. Quite frankly, it's one of my favorite parts because I love doing what I'm doing, being able to teach and work with students, as well as being in the ocean is my ultimate life goal. What about you, Bolt?
Yeah, I'm actually on the operating CCMI. I'm usually driving a boat or fixing one of the trucks. But today I get to take a break and talk to all of you all. We've got others on the boat joining us, both from the operations and education and outreach teams. It's a great group here at CCMI. And likewise, underwater, our camera persons are from the outreach and even research teams today. So, a wide variety of things here at CCMI. We have a shout out from Shane Delpeche from South Africa. I think Chandra would know who that is. So these are all some excellent questions and comments here. Here's a question. Is the lionfish population stable or increasing / declining? From what I've seen, dramatic changes in it recently, unless you know of anything, Chandra.
No, I've definitely seen less and less lionfish. I haven't seen a lot of them. However, there are still a few around, but not in damaging ways.
That's good to hear. I have one more question. Is the bleaching worsening on Little Cayman? To which I would say it comes and goes with the hot summer temperatures. We lost so much coral in that heat wave in 2023 that actually took out a lot of the heat-sensitive coral. Granted, what is left is more heat-tolerant, so that provides, it makes the bleaching events slightly less dramatic than they would be otherwise. But again, that's something that's going to totally depend on the temperatures of the states at a high temperature. I think about marine conservation today. I'm trying to look for any other questions or comments in the chat. Hi from Cindy Rickelli, watching from her kitchen in South Carolina. Loves learning about our seas. Thank you all for joining us. We've spoken a lot about marine conservation today, and there are many opportunities to get involved in marine conservation. You might think about things like supporting research organizations, participating in citizen science projects, or for that matter, even starting local conservation efforts. And I hope you all feel motivated to pursue some of these options and to generate more ideas of your own. Unfortunately, we are getting to the end of our time for today. So before we sign off, let's take a moment to recap what we've discussed in this episode. Chandra, could you give us a brief summary?
Of course. So, we've discussed the importance of seamounts and the role they play in our environment. We also got an introduction to Expedition Hope, which was one of the major groundbreaking research projects that our sea team got to do. We learned about how seamounts affect ocean mixing and how it can potentially be a buffer for climate change. And we also learned about the biodiversity on seamounts.
Great, thanks for that, Chandra. Now we hope all of you enjoyed today's episode and feel inspired to become the next generation of environmental stewards. So thanks for watching, everyone. Chandra, would you care to say farewell to our viewers?
Of course, I'd love to. Hi everybody, thank you for joining, and we hope to see you for the second episode where we'll be discussing